Hello, dear students. Good morning to all of you. Myself, I am Dr. A. Rangrao and my colleague Dr. K. Shrikant. So we are both working as associate professor in the Department of Biotechnology, Vignan's Foundation for Science, Technology and Research. So we are going to deal biochemistry for section A and section B students. So previous topic we learned about carbohydrates, classification and structures. So today I am going to deal about polysaccharides. What is polysaccharide and what are the functions of polysaccharides? So let me discuss about this one today. So this topic covers polysaccharides. So how many types of polysaccharides? There are homopolysaccharides and heteropolysaccharides. Complex carbohydrate, structure of starch and glycogen and structure of dextron, cellulose, glycoproteins and glycoaminoglycans. Generally we call this one GAGS. Anomerism, isomerism, beta rotation. So these are the topics we are going to cover in this topic. Introduction, polysaccharides. Polysaccharides are complex carbohydrates. What is carbohydrate? Previous lecture we learned about carbohydrates. Carbohydrates is made up of carbon, hydrogen, oxygen. So polysaccharides, complex carbohydrates composed of 10 up to several monosaccharides arranged in chains. The most common monosaccharides that appears part of polysaccharides. That means polysaccharides contain monosaccharides. Monosaccharides. Several monosaccharides made up of the polysaccharides. Polysaccharides, polymer of monosaccharides, as I mentioned that, and these are the polysaccharides. Classification according to the biological role. Polysaccharide, storage polysaccharides, and structural polysaccharides. What is the example for storage polysaccharide? Storage polysaccharides example: starch and glycogen. Starch and then glycogen. Coming to structural polysaccharides, cellulose and then chitin. So classification according to number of monosaccharides. Again derivative homoglycans, heteroglycans. Based on that, homoglycans and then heteroglycans. Properties of polysaccharides. What are the properties of polysaccharides? So they are sweet taste. They are amorphous substrates in soluble water. They are soluble in water. They are insoluble in water. They have high molecular weight. The molecular weight is very high, and they form colloidal forms in solution. When they dissolve in the solution, they form colloidal form when you heat it with the water, and they do not exhibit any properties of aldehyde and ketone group. Aldehyde, ketone. What is aldehyde and ketone group? They are actually carbonyl groups. They are in carbonyl groups. So they don't exhibit any properties of aldehyde and ketone groups. Homopolysaccharide. What is homopolysaccharides? They contain monosaccharides units of single type. They contain monosaccharides units of single type. Example: glycogen and starch and cellulose. These are some of the examples of homopolysaccharides. Starch and then glycogen, cellulose. These are some of the examples. The polymer of alpha glucose. This is very, very important. Alpha glucose. There is starch. The polymer of alpha glucose is starch. Starch. How the starch forms? Alpha glucose. They are a polymer of alpha glucose, starch. The polymer of beta glucose is cellulose. This is the cellulose. They form beta glucose. Heteropolysaccharides. They possess two or more types they have two or more types. There are two or more types monosaccharides. Two or more types of monosaccharides. Is the derivatives. Example, heparin. Example, you can take heparin. Okay. You can see the heparin structure here. So sulfated glucosamine. Sulfated glucosamine. You can see the structure here. This and this. These are the sugar molecules here. And glucose looks like a glucose structure. And amine group attached to the amine group. This is carbon 1 and this is carbon 4. Okay. So they are attached together. Here this is a bond, sulfated bond. You can see the sulfated bond here. So this is the sulfated hydrochloric acid and this one is sulfated glucosamine. Sulfated glucosamine. So this is a heparin. This is a heparin structure. Example, heteropolysaccharides. Complex carbohydrate. They are made up of sugar molecules that are stretched together in a long chain. They are in a long chain. So that's what they are mentioned that complex carbohydrate. Very long chain and they contain sugar molecule. They attach it to the one sugar molecule. They are very long chain. So they are found in foods such as peas and beans, whole grains and vegetables. You can see such a carbohydrates in natural sources. From the natural sources, you can see this type of complex carbohydrates. You can able to see in the foods, peas and beans and whole grains and vegetables. Both simple and complex carbohydrates are turned together, glucose, blood sugar in the body and are used as an energy. Why we are using carbohydrates? We are using carbohydrates for an energy purpose. So we need a sufficient energy. Nowadays, we don't have a sufficient energy, so that's what we are use more carbohydrates, then you will get more energy. Example, okay. So both simple and then complex carbohydrates are turned to glucose and blood sugar into the body and used as an energy purpose. We will keep the body fuel for extended period of time. If you use this type of this one, you can get the fuel and you can say extended period of energy you will get to the body. And glycogen. What is glycogen? Glycogen usually found in the animals. This is a carbohydrate. So glycogen is a homopolysaccharide. This is a homopolysaccharides. Homopolysaccharides. This is a homopolysaccharides. Remember that it gives glucose on complex. When you do the complete hydrolysis process, you can get glucose. This is a very important glucose. Glycogen is when you do this is a homopolysaccharides, and when you do this one hydrolysis process, you will get this compound. That name is glucose. It is a main storage carbohydrate in animals. You can found in the animals, liver and muscle. It has a similar structure of amylopectin. The structure is similar to amylopectin. Glycogen structure is similar to amylopectin. It is called as an animal starch. This we can call as an animal starch. Glycogen found in animals in liver and muscle, and the structure is similar to amylopectin. We call it is an animal starch. Glycogen. This is the structure of glycogen. You can see these are the sugar molecules. You can see here one sugar unit, 2, 3, 4, 6. You can see this is a long chain and this is a branched chain. When it is like this is a branch. These are the branched chains. Okay. This is the linkages. As we know, previous class we learned that glycosidic bond. These are the bonds, glycosidic bond between the two sugar molecules. So it will form between the glycosidic bond. Through glycosidic bond, this will form, and you can see glycogen how the structures linked to one sugar molecule to another sugar molecule. And this is a long chain, and this is a branched chain. This is the linkages. Linkages always it forms hemi-acetal or hemi-ketone, between hemi-acetal and between hemi-ketone. So starch. What is starch? Starch is a homopolysaccharide. This is a homopolysaccharide. It's a non-sugar molecule, because when you take this one starch, you know that is not a sweet. Okay, taste. This is not a sweet. So a polysaccharide, homopolysaccharide, a sugar. This is a non-sugar. Starch is non-sugar. it is glucose on complete hydrolysis. When you do hydrolysis process, you will get glucose. You will get glucose. When you do hydrolysis process, you will get the glucose. So it is the main storage material in plants. You can see the starch usually found in the animals. Plants. So whereas glycogen you can see in the animals, but whereas starch you can see in the plants. Most of the time you can see this in the plants. You can see this in the plants. This is starch is the main storage material in the plants. Starch is insoluble. This is insoluble and complete, but it is easily available. You can get easily available for use by the plants. So starch is a storage product in the plants. It is white amorphous. This is you can see it's a white powder. You can see white powder, tasteless and soft substances. You can when you touch this one, it is very soft. So it is a made up of glucose units. This is made up of glucose units. This is glucose. Okay. The glucose units are linked by alpha 1,4 glycosidic bond. Alpha 1,4 glycosidic linkages and branch are formed alpha 1,6. Alpha 1,4 and then alpha 1,6 linkages through the glycosidic bond. Through the glycosidic bond, it will burn. They are sugar units. They actually link to the one sugar molecule and one sugar molecule, but the linkage is alpha 1,4 and then alpha 1,6. Starch consists of two main compounds. This is another main important point you need to remember always. The starch contains two main components. That one is amylose. This is one amylose and another one is amylopectin. These are the two molecules it contains starch. Amylose contains 20%. This starch, okay? So amylase contains 20%, okay? If you see the starch, amylase 20% and the remaining 80% amylopectin. So coming to amylase, it is made up of a single chain of glucose molecule that form spiral. This is a sugar molecule, glucose. Okay, it is made up of glucose molecules with a form of spiral. You can see the spiral, okay, spiral. So amylase is 20%. So amylase 20% containing starch, and then amylopectin is 80%. It contains starch contain 20%. Okay, amylose and then 80% amylopectin. So this is the starch and there are two major components. And the structure, if you see, structure looks like this. You can see here alpha 1,4 linkages. As I mentioned that the starch contains alpha 1,4 linkages, alpha 1,6 linkage. This is the linkages. It will form linkages between linkages. Okay, this is amylose. Amylose only alpha 1,4. If you see amylose structure, you can see the bonds here. You can see each bonds here. Okay, so they are formed alpha 1,4 linkages. Alpha 1,4 linkages. Whereas amylopectin you can see here, they form alpha 1,4 and alpha 1,6 bonds. You can see glycosidic bond. So this is the starch. Starch contains two major components. One is amylase, another one is amylopectin. Amylase 20%, this is contain 20%, and this one contain 80%. And dextron. It is a complex branched glucan, composed of chains of varying length. They are very long. Okay, the 3 to 200 kD, 3 to 200 kD. It is used medicinally as an anti-platelet purpose. They use to reduce blood viscosity and as a volume expander. Volume expander in hypovolemia. They use in medicinal purpose. They have medical applications. They use in anti-platelet to reduce blood viscosity. And coming to volume expander, to expand volume. So in hypovolemia they will use. And it is synthesized from sucrose. Sucrose by certain lactic acid bacteria, the best known being a leucoconstituent and then some of the mutants. So these are the bacteria they used, and then they synthesize this one. It is synthesized sucrose through the sucrose by lactic acid bacteria. We have provided some of the examples here. You can look later. And the straight chain consists of alpha 1, 6 linkage. The straight chain consists of alpha 1, 6 glycosidic linkages between glucose molecules. This is the alpha 1, 6 linkages. They are alpha 1, 6 linkages. You can see here alpha 1, linkages between glucose molecules, while branches begin from alpha 1, 3 linkages. When the branches begins from here, this is alpha 1, 3. Alpha 1, 3 linkages, but here is alpha 1, 6 linkages. The straight chain consists of alpha 1, 6. These are alpha 1, 6. This is all alpha 1, 6 linkages. Whereas this one alpha 1, 3 linkages. Cellulose. Coming to cellulose, it is a non-sugar and homopolysaccharide. Cellulose is a non-sugar and homopolysaccharide. It is composed of glucose units, polymer of beta glucose. This is very, very important. Polymer of beta glucose. This is very, very important. This is it will form cellulose, polymer of beta glucose. And this is the structure. Each monomer is inverted. Each monomer is inverted as a consequence for its properties. So, cellulose is a non-sugar homopolysaccharide. It is composed of sugar units and it will form polymer of beta glucose units. Polymer of beta glucose units. Each monomer is inverted and as a consequence of its properties. You can see here cellulose. This is a cellulose. You can see here this is a cellulose. Okay, and beta 1, 4. This is a, you can see beta 1, 4 glycosidic bond. Beta 1, 4 glycosidic bond. They have a linkage beta 1, 4 glycosidic bond. It is not easily hydrolyzed. It is not that much easy to hydrolyze. So it is hydrolyzed to glucose by concentrated H2SO4. Either we need to use acid, okay? We need to use acid and NaOH, alkaline solution. Okay, we need to prepare this one and we need to use when we do the hydrolysis process. But it is not that much easy because the cellulose is very, very thin. So a very, very big one molecule if you want to break down. So it is hydrolyzed into glucose by concentration H2SO4 and then NaOH. You need to use this one. And it is maintained consequent of cell walls of plants. You can able to see this one, cell walls of the plant. Cellulose form cell walls of the plants. You can extract from the cell walls of the plant. It occurs in lignin and cotton. It occurs in lignin and cotton. Its molecular weight is very big. The molecular weight is big. You can see here mentioned. It contains 1200 to 12,500 glucose units. Very big one. So one glucose unit form another and another it will form. So these are actually contain. It contains more than 1200 to 12,500. It's a very big one. The sugar molecule contains very, very big one. So molecule, one molecule of this one, cellulose, it contains 1200 to 12,500 sugar units. Glycoproteins. Coming to glycoproteins. What are glycoproteins? Glycoproteins are proteins that contain oligosaccharides. Oligosaccharides that contain oligosaccharides chains. Oligosaccharides and covalently attached. They are covalently attached to polypeptide side chains. They are covalently attached to polypeptide side chain. So glycoproteins are proteins contain oligosaccharide chains. Oligosaccharides is nothing but glycans covalently attached to the polypeptide side chain. Polypeptide side chain. The carbohydrate is attached to the protein in co-translational post-translational modifications. Either co-translational or post-translational modification. This process is known as a glycosylation. This whole process we call glycosylation. So glycoproteins are very important. Integral membrane proteins. Glycoproteins are very important. The internal membrane proteins where they play an important role. Cell-cell interactions. Cell-cell interaction they play a major role. So these are glycoproteins. There are two types. Glycosylation. Okay, there are two types of glycosylation. So one is N-glycosylation. N means nitrogen glycosylation, and the other one is oxygen glycosylation. So N-glycosylation, the addition of sugar chains can happen at the amide nitrogen, on the side chain of the aspargine. What is aspargine? Aspargine is an amino acid. So N-nitrogen glycosylation. The addition of sugar chains. Can happen the amide nitrogen on the side chain of the aspargine. Aspargine is an amino acid. And then oxygen glycosylation. The addition of sugar chains can happen on hydroxyl oxygen on the side chain of hydroxylysine, hydroxyproline, serine, threonine. These are some of the amino acids. This one, and this one. So oxygen glycosylation happen. The addition of sugar chains can happen on the hydroxyl oxygen on the side chain of these are the amino acids. Hydroxylysine, serine, hydroxyproline, and threonine. So there are two types of glycosylation. So one is nitrogen glycosylation. This one is the addition of sugar units can happen amide nitrogen on the side chain of aspargine. Aspargine is an amino acid. And oxygen glycosylation. The addition of sugar units attached to can happen hydroxyl oxygen on the side chain of amino acids. Those amino acids is hydroxylysine and then hydroxyproline and then serine, threonine. So these are the difference in glycosylation and then oxygen glycosylation. This is structure here, oxygen and then nitrogen linked. How they link to the amino acids? So glycoproteins. So aspargine. Okay, you can see this is the linkage, N linked and then oxygen linked. Okay, this is N-acetyl glucosamine. You can see here attached to here, and then we can see this is the aspargine amino acids and linked to the amide. Okay, this is polypeptide backbone. This is a N-linked, nitrogen linked N-glycosylation. Coming to oxygen glycosylation. This is the serine amino acid and threonine, either serine and then threonine. This is the amino acids linked to oxygen. Okay, N-acetyl galactosamine. This is a N-acetyl glucosamine, and here N-acetyl galactosamine. So this is a sugar molecule attached to the amino acids. The oxygen, this is amide. So this is oxygen linked. These are the oxygen linked glycosylation. So this is the difference. Glycoproteins. Okay, this is the difference. N-linked and then oxygen-linked glycosylation. And glycoproteins. What is the importance of glycoproteins? Glycoproteins are very important, and for white blood cell recognition, they use white blood recognition purpose. They use especially mammals, such as use cell. So glycoproteins are very important for white blood cell recognition, especially in mammals. Molecules such as antibodies, Ig, okay, immunoglobulin, okay, which interact directly with the antigens or glycoproteins. So they use in this process molecules such as antibodies, you know antibodies, okay, which interact directly with the antigen. So in that antigen are glycoprotein. They actually are glycoprotein. They use these glycoproteins. Okay, molecules of major histocompatibility complex. Okay, which are expressed the surface of cells interact with T cells, interact with the T cell as part of the adaptive immune response. In this process, glycoproteins also used. Again, glycoproteins. Okay, 2B and then 3A are integrant. So found on platelets. These are found on platelets that is required for normal platelet aggregation. Okay, and adherence to the endothelium. Components of the zona pellucida, which surrounds the and it is important for the sperm, okay, egg interaction. So these are some of the properties of glycoproteins. Glycoproteins used for the various purposes. Next one is glycosaminoglycans. Usually people short name GAGS. Okay, glycosaminoglycans. Okay, glycosaminoglycans are long unbranched polysaccharides. Large, they are very big one. So unbranched polysaccharides consist of repeating sugar in it. They contain actual sugar in it. They contain sugar. How the disaccharide will form? Disaccharide when it will form between two sugar molecules when it is happening a condensation process. So that time disaccharide will form. So glycosaminoglycans are very long unbranched polysaccharides. Okay, consist of repeating sugar units. They are repeating disaccharides. The repeating unit consists of amino sugar, N-acetyl glucosamine or N-acetyl galactosamine. These two repeating consist of repeating sugar units. One is nitrogen acetyl glucose, amine, and then N-amino acetyl galactosamine. These are the two consist of along with the uronic sugar, okay, and or galactose. So the repeating unit consists of amino sugar. These are the amino sugars. It contain glycosaminoglycans. Okay, and then glycosaminoglycans are polar. They are very polar and attract water. They are very attracted to the water because they are polar nature. Therefore, useful to the body as a lubricant. We use these actually lubricant purpose or as a shock absorber. So, for this purpose, they use glycosaminoglycan, GAGS. GAGS have a high degree of heterogeneity. They have a high degree of heterogeneity. Okay, with a regards to molecular mass. So, coming to glycosaminoglycans, GAGS. They have high degree of heterogeneity. This is heterogeneity with regards to the molecular mass. Okay, disaccharides construction and sulfation. Because of this one, disaccharides construction and sulfation. Because of this one, they are heterogeneity, a very high degree of heterogeneity based on core disaccharide structures. So GAGs are classified in different. So heparin, example, and chondroitin sulfate, and keratin sulfate, and hyaluronic acid. These are some of the GAGs. These are the classified different four: heparin, chondroitin, keratin sulfate, and hyaluronic acid. Again, this HSGAGs means this one. Okay, heparin sulfate, glycosaminoglycans, heparin sulfate, glycosaminoglycans, and CaGACS, that means this one, chondroitin sulfate, glycosaminoglycans. Okay, keratin sulfate. Okay, synthesized in the Golgi apparatus. Golgi apparatus with oxygen linked and then nitrogen linked. Oxygen nitrogen linked glycosylations as a post translation modification. As a post translation modifications. So hyaluronic acid is synthesized by integral membrane synthesizes which immediately secrete the dynamically elonged disaccharide chains. So these are the GAGs very heterogeneity and actually classified 4 heparin, chondroitin sulfate and then keratin sulfate and then hyaluronic acid. Again these are synthesized in the apparatus, Golgi apparatus, okay, with oxygen and nitrogen linked post-translational modification. These are the structure of chondroitin sulfate. This is the structure of chondroitin sulfate. And then this is the keratin sulfate. This is the keratin sulfate. This one is the chondroitin sulfate. And the next one hyaluronic acid. This is the heparin. This is some structure how they are structurally structured compounds. This is how structurally they are in GAGs. Okay, glycosaminoglycans. These are examples. This is based on the classification. You can read the structures. And functions. What are the functions? Heparin is an anticoagulant. Going to use this heparin anticoagulant. So anticoagulant. They use anticoagulant and heparin sulfate has a numerous biological activities. They have numerous biological properties, functions, including cell adhesion, regulation of cell growth, and proliferation. Okay, regulation of cell growth and then proliferation, and developmental process, cell surface binding, lipoprotein, lipase, and other proteins, angiogenesis, viral infection and tumor metastasis. These are the properties. Heparin sulfate has a numerous biological properties. These are some of the properties we mentioned here. Cell adhesion and regulation, cell growth and then proliferation, developmental process, cell surface binding, binding of lipoprotein and lipase and other proteins, and then dermatic sulfate. This is and this one is the dermatic sulfate interact with the fibroblast. This compound interact with the fibroblast growth factor FGF2 and FGF7 have been implicated in cellular proliferation and wound repair. So keratin sulfate helps in the maintenance of tissue hydration. This will help you. Keratin sulfate helps in the maintenance. It is maintenance of tissue hydration. So hyaluronic acid is a major component of the synovial tissues and fluids as well as other soft tissues and other environment remarkable rheological properties. So this has a hyaluronic acid used as a major component in various rheological properties. So these are the functions of GAGs, glycosaminoglycans. And lectins. Coming to lectins. Lectins are carbohydrate binding proteins. They are carbohydrate binding proteins. They are macromolecules. Micro and then macro. Macromolecules, bigger one. Okay. Lectins are carbohydrates binding proteins. They are macromolecules that are highly specific for sugar molecules. And plant lectins. Examples here. This is plant lectin, phytohemagglutinin, and were noted for their particular high specificity for glycoconjugates. Those are fungi, invertebrates, and animals. These are the and then used biomedicine. These lectins are used in biomedicine applications for blood cell testing. This is lectins used in blood cell testing purpose in biochemistry and fractionation. So they are lectins used in blood cell testing purpose. So lectins are carbohydrate binding proteins and they are macromolecules that are highly specificity for sugar moiety. And plant lectins are also known as a phytohemoglutamins. Phytohemoglutamins. They are particularly high specificity for foreign glycoconjugates. Foreign glycoconjugates. Those are fungi examples in the animals and used in blood cell testing purpose. Okay, coming to lectin, this one, the properties, the functions. But the lectins perform, recognize on the cellular and molecular level lectins. We are talking about the lectins, okay? Lectins. So, this lectins, okay? Lectins perform, recognize on the cellular and the molecular level and play numerous role in the biological recognition. In biological phenomena involving in cells, carbohydrates and proteins. Carbohydrates and then proteins. Okay. So lectins perform recognition on the cellular level and then molecular level. Particularly in the cells, living cells. And lectins mediate attach and binding bacteria and viruses. Lectins also used. These lectins actually used and mediate attachment and binding. They attach and then binding. Okay, so bacteria of viruses. Lectins mediate attachment and binding of bacteria and then viruses to their intended targets. When you do the target particular, then you can do this one. Lectins mediated attached to bacteria and then viruses. Then example, it is hypothesized that some hepatitis C viral glycoproteins attached to C-type lectins on the post cell surface, liver cells for infection. So this is one of the example. It is hypothesized that some of the hepatitis C viral glycoproteins attached to C-type lectins. C-type lectins on the host cell surface. Example, liver cell for infection. Coming to anomalism. What is anomalism? So carbon 1 position. If you see the glucose structure, we know glucose C6H12O6. So carbon number 6 carbon. Glucose contains 6 carbons. Okay. Carbon 1 position. The cyclic structure is called anomeric carbon. Carbon 1 position. Okay. Carbon 1 in the cyclic structure. If you see the cyclic structure, carbon 1 is called anomeric carbon. Anomeric carbon. It is a asymmetric carbon atom. Symmetric and asymmetric. Okay. It is a symmetric carbon atom. It is a symmetric carbon atom. If the OH hydroxyl group at C1, the anomeric carbon is to the right side, it is called alpha form. This is the alpha form. If the OH attached C1, if the OH is attached to the carbon 1 position, to the right side, if it is the right side, attached to the right side, it is called alpha form. It is called alpha form. If the OH attached C1, is to the left side, it is the right side and the left side called beta form. This is actually beta form. Beta form. So this you need to remember that anomeric is a cyclic structure. The carbon 1 position in the cyclic structure is called anomeric carbon. But here you need to remember that OH attached to C1, if it is the right side, it is alpha position. If the OH attached to C1, attached to C1 at the left side, that we can say that beta form. You need to remember that alpha form and then beta form. Anomeric. What is anomerism? This is the one you can see here, alpha D glucose. This one is alpha D glucose. This is alpha D glucose. You can see here carbon 1 position. This is a carbon 1 position.
This is the OH attached here. Okay, attached. Okay, so that's what we mentioned here, alpha D glucose. And whereas we can see the left side here, this one we can see left side, so that we can mention here beta glucose. So alpha and beta forms of sugars are called anomers. They are anomers, type of stereoisomerism. They are a type of stereoisomerism. So you need to remember that anomerism means the OH is attached to right side, alpha. If the left side, the OH is attached to the left side. What is isomerism? Isomerism chemically have similar, they are a same molecular form, they are same molecular formula and different structural stereochemical formula. Isomers are chemical compounds. They are chemical compounds, but they are having same molecular formula but different structural stereochemical. And functional groups. Isomerism. Functional group is isomerism. Aldose or keto isomerism. Functional groups. When I talk about the functional group, functional isomerism. Functional isomerism means there are, one is aldose and the next one is keto isomerism. Aldose isomerism or keto isomerism. That is functional isomerism. So glucose or fructose are aldo-ketose isomers. They are glucose and then fructose or aldose keto isomers. They are aldose keto isomers. Glyceraldehyde and dihydroxyacetone are aldose ketose isomers. Glyceraldehyde. If you see the glyceraldehyde structure, at the end of the C1 position you can see CH2. And whereas this is the C1. But whereas this is the glyceraldehyde. If you see the dihydroxyacetone, you can see the C and C. Here is the second position contain ketone. So that's what these are the functional isomerism. Functional means aldose and keto isomers. These are example. Glucose and fructose. Okay, both are aldose and keto isomers. Examples: glyceraldehyde, and the next one is dihydroxy or the aldo keto isomers. Mutarotation. What is mutarotation? Mutarotation, always need to remember the interconversion of alpha and beta. Interconversion of alpha and beta anomers. Okay. Interconversion of alpha and beta we call mutarotation. That we call interconversion alpha and beta. We call that is mutarotation. So alpha and beta anomers, both are anomers of carbohydrates, typically stable solids. They are stable solids. In aqueous solution, if you see in the aqueous solution, so they are quickly equilibrate to an equilibrium mixture of two forms. They will form immediately two forms. Okay. That when you do this one solution, glucose exit 36% of alpha, 64% of beta. So greater than 90% of pyranose forms exist in solution. It is 60% of beta. 60% of beta. Greater than 99% of this one pyranose forms exist in solution. So this is beta rotation. Beta rotation is interconversion of alpha and beta. Okay, alpha beta. Alpha and beta anomers of carbohydrates, typically they are stable solids. When you dissolve this in the aqueous solution, then it will form two molecules. So 36% of alpha and then 64% of the beta. But the greater than 90% of pyranose forms exist. These are the summary questions. This is the possible test questions. You can have a look and explain detail about the chemical forms of polysaccharides, and then what are polysaccharides, and describe structure and properties of polysaccharides, and discuss about homopolysaccharides and then heteropolysaccharides. Write note difference between amylose and amylopectin. It is very, very important. Amylose and amylopectin, both are in starch. And discuss about some of the structures, starch and then glycogen and then dextron, and then explain cellulose, chitin and then GAGS, glycosaminoglycans and lectins. Discuss about the polysaccharides helpful in the formation of plants and animals. Okay, so write about GLGS and glycoproteins. These are some of the important test and these are some of the books, textbooks and reference books. You can read this one, you can gain more knowledge, and you can thorough what these are the structures, how they link to the sugar molecule and how they are actually structurally, and you can gain more knowledge. So I request all of you to read these books. When you go to the library and read more of this. It is available in the